Okay, so in chapter 12, we're going to talk about HIV disease and AIDS. So, HIV and AIDS. AIDS is an acquired immunodeficiency disorder that results from defective immune functioning, and AIDS comes about from HIV infection. So, really, AIDS is the chronic consequence of HIV infection. Now, what's hallmark about HIV and AIDS is you see a defective cell mediated immunity where there's a decrease in CD4 positive T helper cells. Now, normally these T helper cells will release cytokines to activate other immune cells, but in the case of HIV and AIDS, it turns out that B cells and T cells lack that sort of communication, so there's a dysfunction of the immune system as a result, and thus the immunodeficiency that results. Now, in terms of the epidemiology of HIV and AIDS, there are approximately 33 million people living with AIDS worldwide. Each year, there are about 1.8 million AIDS deaths, and most of these AIDS cases are in sub-Saharan Africa. However, it is spreading in other parts of the world, like Asia and Eastern Europe. In the U.S., there are approximately 663,000 people living with HIV and AIDS, and approximately 56 or so thousand new HIV infections every year. So the prevalence is about 600 or 700,000, and the incidence is about 60,000 or so new HIV infections every year. Now, most of those new HIV infections occur in men, and of those, about half occur in men who have sex with men, and 90% of children younger than the age of 13 were infected perinatally. So, sometime around birth, that's when HIV was sort of introduced to their body. Now, there are different isoforms or subtypes of HIV. One type is HIV-1, and it's thought to have originated in Central Africa. Most cases of AIDS are found in Central Africa, United States, Europe, or Australia, and there are probably about 10 subtypes of HIV-1. For HIV-2, it's thought to have originated in West Africa. It's found primarily in West Africa, and it's characterized by a longer latency and asymptomatic period and a milder form of the disease, and therefore lower mortality rates. However, like HIV-1, HIV-2 can also progress to AIDS, which is acquired immunodeficiency syndrome. Now, for transmission, there are really just three main types of transmission of HIV. The first type is sexual transmission. So it could be through semen or vaginal secretions, homosexual, bisexual, or heterosexual intercourse. And another subtype would be parenteral transmission through blood or bloodborne products, blood-contaminated needles, that kind of stuff. Or perinatal transmission in utero, during delivery, or in breast milk. HIV can be transmitted from mother to infant in that way. Now, in terms of the structure of this virus, it consists of a core nucleocapsid that contains two RNA strands. So the RNA genome of HIV, there's two copies here. You have a protein capsid here and a lipid bilayer envelope. Now, on this lipid bilayer envelope, we find lots of glycoprotein spikes like GP41, GP120, and these glycoprotein spikes are necessary for adhesion of the virion here to its host cell. So if you block these glycoprotein spikes, that's another potential pharmacological target to prevent the adhesion of the virus to a host cell. If the virus can't bind and enter, then you won't get an infection. Now, one of the virulence factors that's produced by HIV is something called reverse transcriptase. Now, remember, HIV's genome is in the RNA form, and what reverse transcriptase does is that it converts RNA into DNA. Now, once the virion has entered its host cell, this reverse transcriptase will copy the RNA viral genome into a DNA genome. And what the purpose of this is that the DNA genome of HIV is then inserted into the host cell genome through an enzyme called integrase. Now, we also have another enzyme that's brought in by the HIV virion called protease. These are essentially molecular signal scissors that split viral components by autocatalysis, and they clip different proteins to make it infectious. So a typical HIV life cycle looks like this. So pre-infection, and post-infection. And what you end up with then is a basically identical copy of the virus that initially infected. And the way this works then is the HIV virion will bind to and fuse with a host cell. And this is necessary through the action of those glycoprotein spikes. So like GP41, GP120, those are used to bind to the host cell genome. Upon binding of the virion to the host cell membrane here, it injects its viral RNA into the host cell. Now this viral RNA is then converted to DNA by the action of reverse transcriptase, and then the DNA genome migrates over to the nucleus, where it's integrated randomly at some point somewhere in the genome of the host cell through an enzyme called integrase. So integrase actually will integrate the DNA copy of the HIV genome into the host cell DNA in a random spot. Sometimes this kills the host cell, sometimes it doesn't, and sometimes it also induces cancer. So there's a rare chance that HIV will insert its genome in a place that mutates a gene enough to make a host cell cancer. So there are some forms of lymphomas that are due to HIV infection. Now, once the host genome is integrated into, once the virus genome is integrated in the host genome, it basically hijacks the host cell's machinery to make messenger RNA, which is used to make viral proteins. And so basically at this point, you're using the host's ribosomes and you're using other host molecules to make viral proteins. And you can also copy the DNA genome of the virus into an RNA copy. Now all of this is assembled into new virions, which essentially bud out of the host cell. You might wonder, well, why is it important to know the life cycle of HIV? Well, any of these aspects of the HIV life cycle are all potential targets for different forms of drug therapy. So let's say, what if you can prevent the fusion of the virion with the host cell? Well, then it could never inject its genome into a host cell and you couldn't get an infection. Or what if you blocked reverse transcriptase? Well, then you couldn't get RNA viral genome being copied into DNA, which means it can never integrate and complete its life cycle. Or what if you prevented the production of viral proteins, or prevented the protease function here that is necessary for viral proteins to be cut up and assembled into new HIV virions? Well, these are all potential pharmacological targets, and we actually do have drugs to block many of these aspects of the HIV life cycle. Now, in terms of the progression of HIV, it's first introduced to a host through a variety of mechanisms, but let's just say that it's first introduced through this mucous membrane. If it crosses that mucous membrane through like a micro tear or some sort of wound, then they can enter the tissue. Once this HIV virion is deep in the tissue, it'll bind to cells that are CD4 positive. So some examples of CD4 positive cells would be things like T helper cells or dendritic cells. And HIV will then infect T helper and dendritic cells. Now, dendritic cells are a mobile type of macrophage, and these cells can walk and spread throughout the body. Also, T helper cells will travel throughout the lymphatic system. And once inside these cells, basically the virus will piggyback onto the movement of these cells and spread throughout the body. You'll find then that it can infect lymph nodes and spread to more cells in that local area. Once you get viremia, the virus can spread to other parts of your body through the bloodstream, whereby it can affect more distant cells. Now, what will happen eventually, you guys, is that a patient with HIV infection can enter clinical latency. And it's a period of time where there's no signs or symptoms of HIV infection. But this doesn't mean that someone's HIV infection is cleared. It's possible that the virus is just dormant in the genome of one of their immune cells. Now, other microbial infections or cytokines can actually increase viral replication, and the virus can surge back when it kind of can determine if there's a sort of an opportunistic time to reinfect. Now, upon multiple sort of relapses and latencies, over time, this can lead to AIDS. Now, AIDS is a period of time, and it's a clinical definition, where there's a destruction and depletion of these T helper CD4 cells. And there's a particular lab value cutoff where it's the number of CD4 cells per microliter of blood that you now technically enter AIDS. Now, because of these individuals having decreased immune functions, they end up being more susceptible to opportunistic infections, infections like herpes zoster. You can see here the herpes sores from like HSV1 or HSV2 in the oral mucosa of this gentleman. Another example of an opportunistic infection that results from HIV are infections of Epstein-Barr or human papillomavirus, which can lead to things like oral hairy leukoplakia, which are basically like small kind of hairy shaped plaques that can show up in the oral cavity here. Another form of an opportunistic infection is Candida albicans, which is basically yeast. So this is a fungal infection, and you can see that this sort of swollen tongue and whitish appearance of the tongue is due to a yeast infection of the oral mucosa. Where Candida albicans is normally part of our microbial flora, if the immune system is suppressed, then it can have an opportunistic infection of like the oral or genitourinary mucosas. Now, there are some neurological manifestations of HIV as well, because neurons are also CD4 positive. And this means that HIV can infect neurons. So over time, what can progress then is HIV encephalopathy. We also call this AIDS dementia. It's a complex subacute form of a neurological disorder that can lead to really complex neurological manifestations, and it can be caused directly or indirectly by the HIV infection, viral products, or cytokine-related cellular damage. And this is essentially a form of dementia that's caused by AIDS itself. And so over time, people with a chronic HIV infection or living a long time with AIDS, they can develop a form of dementia that's due to their infection. Now, another form of manifestation of HIV is something called Kaposi sarcoma. It's a common tumor found in HIV-infected men, and it's a form of skin lesion where you find these individual tumors that are benign, flat, macular, which means they're kind of raised. They're subcutaneous, and they're going to range from light pink to a deep purple. They're painless, non-blanching, and non-pruritic, so they don't itch, and they're not painless. What this looks like then are these. So these sort of pigmented skin eruptions that don't hurt, and they're raised, but they also don't itch. It's a form of a tumor that results from HIV infection, and it's a common skin eruption called Kaposi sarcoma, and it's due to HIV. Now, in terms of treatment of HIV, what we want to do is basically target all those aspects of the viral life cycle. So we call this antiretroviral therapy. You can have reverse transcriptase that stops the conversion of RNA to DNA. You can actually have integrase inhibitors, which prevent the DNA viral genome from being inserted or integrated into the host cell genome. You can actually block the fusion of the HIV virions to a host cell, or even have protease inhibitors, which prevent viral proteins from properly assembling. And if all of these work correctly and in conjunction, what you'll find then is that the viral life cycle can't complete. So whereas you might have had a normal viral life cycle producing copies of virions, you can see that no effective copies are over here. And antiretroviral therapies have progressed pretty well over the last 30 years or so, to the point where HIV infections are no longer a death sentence. There are people who live, decades and possibly full, happy lives, even being infected with HIV because they have it under control with their antiretroviral therapies.